My name is Alyssa Arias, and today I'm going to be talking about protein synthesis. What is protein synthesis? Protein synthesis is the process where cells make proteins, and it happens in two stages, transcription and translation. Transcription is the transfer of genetic information in DNA to mRNA in the nucleus, and translation is the process of translating the sequence of a messenger RNA, which is mRNA, molecules to a sequence of amino acids during protein synthesis. Now, where does protein synthesis occur? Protein synthesis happens in cellular structures called ribosomes, which are found outside the nucleus, and during transcription, a strand of RNA is synthesized. What is the function of protein synthesis? Protein synthesis is the process where all cells use to make proteins, and these are responsible for the cell structure and function. What is required for protein synthesis? The three roles of RNA in protein synthesis, which are messenger RNA, which is translated into the protein by a joint action of tRNA, and the ribosome, which is composed of different kinds of proteins and two major ribosomes, rRNA molecules. rRNA associates with a set of proteins which form the ribosomes. The steps of protein synthesis. There are 4 steps, and the first step is the transcription of mRNA from a DNA gene inside of a nucleus. Before this, all the other types of RNA have to be synthesized using the appropriate DNA. The RNAs migrate from the nucleus to the cytoplasm, so before beginning all of this, the component parts are already assembled in the ribosome. The second step is initiation. In the cytoplasm, the protein synthesis is actually initiated by the AUG codon on mRNA. The AUG codon signals that both of the ribosomes with mRNA and tRNA are with the codon AUC since they go backwards. The tRNA initiates the protein synthesis, which has the N-formylmethionine attached to it, which is the next step for the second tRNA to approach the mRNA, which the codon is CCG. This code is the code for proline, and the anticodon of proline, tRNA, reads is GGC. The final process has to start growing the peptide chain by having amine proline to bond the carboxyl acid group, which is MET, in order to elongate the peptide. The third step is elongation. Elongation of the peptide happens because various tRNAs read the next codon. When the correct match is found with the anticodons of tRNA, the tyrosine forms a bond of peptide where the growing peptide chain, the proline is now hydrolyzed from the tRNA, and the proline tRNA moves away from the ribosome and back into the cytoplasm so they can reattach and do another proline amino acid. The last step is termination. Termination is when the stop signal on the mRNA is reached and the protein synthesis is terminated and the last amino acid is hydrolyzed to form the tRNA. The peptide chain then leaves the ribosome and the M-formylmethionine that was used to initiate the protein synthesis is hydrolyzed and it's completed the peptide at its time, and the ribosome is now ready to repeat the synthesis several more times. And that's my video on protein synthesis.